Good evening, ladies and gentlemen. My name's Gary Egan. I'm the Director of Monash Biomedical Imaging, and welcome to MBI and to this ICT for Life Sciences Forum event. I wanted to tell you at the start here that the ICT for Life Sciences Forum is now a very well-established forum over the past 4 or 5 years, and I think it's really a tribute to Lawan and to the ICT and the NICTA team that they've established such a forum as we have here. I mean, it's focused on the convergence of life sciences with mathematics, engineering, IT, and over the last 4 years or so, it has really brought together disparate groups that I think would have otherwise probably not yet sat around a table together if it hadn't been for this fora and for the events that go associated with it. I moved here to Monash 2 years ago, and at that time I thought it would be very important to have Monash University and Monash Biomedical Imaging as a sponsor of the forum, and so we have done that, and we're continuing that through 2013. It's something that we're now hosting here at Monash for the first time, so we're delighted with the turnout that we've got, and we hope that we'll be able to have further fora here in later this year and next year. So it will now, I guess, bridge across the real strengths in the ICT area and the biomedical sciences area across Monash and Melbourne universities and the other institutes that are affiliated with the fora. So from my point of view, I think it's a really terrific evening that we've had the first event here at Monash. Now, with that introduction, I'd like to turn to our guest speaker tonight, Dr. Allan Jones, who's a visitor here to Melbourne at the moment, associated with the launch of the International Neuroinformatics Coordinating Facility. And I'm really delighted that Allan was able to fit us into his busy travel and work schedule. We're really thrilled that you could come, Allan, and thank you for that. Allan has got a really amazing bio, and I'd like to read a little bit from his website just to give you a sense of what Allan's background is. He's got a broad scientific experience in genetics, molecular biology and development. He's got a BSc degree in biology from Duke University, a PhD in genetics and developmental biology from Washington University School of Medicine. Allan joined the Allan Institute for Brain Research, which I think at that time was probably the Allan Brain Atlas facility or research centre, and it's now recently been expanded. But he joined there in 2003 as one of the very first employees. And he brought to the Allan Institute his project leadership, particularly in high throughput genomics, from his prior management positions at Merck, Rosetta in Pharmaceutics, Avitec Diagnostics. And then Allan was particularly instrumental and responsible for building up the multidisciplinary team, which the Allan, I guess, has now really in the vanguard in world research in neuroinformatics, and in this convergent space between ICT and neuroscience. So it's particularly great to have you here this evening, Allan, given the basis of our forum. Allan continues to work with the founders of the Allan Brain Institute, the scientific advisors and business advisors, and has had a lead role in the large-scale and high-impact initiatives from the Mouse Brain Atlas through to the current work on the human brain, which I think we'll hear about more tonight. Another very interesting aspect of the way that Allan led the development of the Allan Brain Institute was the business model that has underpinned the research activity, whereby the projects from conception through to delivery have been basically free public resources which have progressively gained support from the NIH after being initially funded through a major grant from Paul Allen, the founder of the Institute. And so it's delightful to have you here this evening. Thanks, Allan. And welcome you up to the podium to give the address.
Well, thank you, Gary, so much for that lovely introduction. I should say I was speaking to a reporter from the Australian a couple of days ago, and he said, there's another Allan Jones here who is a local shock jock, and so for those of you who showed up expecting the other Allan Jones, I sincerely apologize. I'm not going to offend tonight, at least I hope not to. So let me talk a little bit about the human brain. So when you think of the word complexity, one of the first things you can imagine thinking of is the human brain. And for centuries, we as humans have been fascinated by the brain. And we've used the tools of the day to try to study and understand it, from pen and paper from Da Vinci's time to the current amazing set of tools that you can see right here at the Monash Biomedical Imaging Center, which allow you to see all kinds of details about this fascinating structure. So when you first look at a real, fresh human brain, which this is, one of the first things that will strike you is how bloody it is. There's so much vasculature, there's so many veins that are serving this organ. I like to call this metabolically voracious organ. So something on the order of a quarter of all of the oxygen that is being used by your blood, something like a fifth of the glucose that's used by your body is consumed by this organ. Researchers in the late 20th century learned to take advantage of the fact that this is such a metabolically active organ, and where blood is being consumed and oxygen being used, you get a signal. And that allowed people to start to look at the brain and see what areas were being turned on in a variety of different activities. So the area we're looking at here is sort of temporal cortex on the side, which would be like auditory processing. You're hearing what I'm saying now. Coming around to the front of the brain is where this is your sort of executive function, the areas where you're making decisions, you're doing sort of higher order processing of information. And these kinds of resources are the cornerstone of a lot of brain research, is trying to understand areas that are being turned on. They tell you a lot about the where, but we're very interested in the what. And so when you look at a slab of this brain, it doesn't look like there's a lot of structure. But in the late 1800s, some chemists learned how to stain cells. This is a Nissl stain. It's a light purple stain. And so it's staining the bodies of neurons within this. And you can see that it's not uniform stain at all. So there are whiter areas, and then there are more purple areas. And the purple areas are areas where there's a lot of cells that are being stained. So in a given human brain, there are about 86 billion of these neurons. Some of the more interesting parts are the outer wrinkly parts. There's a 6-layer structure called the neocortex, which is arguably what makes us uniquely human. We have more of it than any other animal. We have so much of it that it can't all fit in our skull without being wrinkled up like it is. And those central processing units really are the things that make us uniquely human. So if we fly in a little bit, let's look at an area. This is an area called the hippocampus, part of processes in learning and memory. And you can see individual neurons here. So these are really the functioning units of the brain. They are wired up. Each neuron itself can be connected up to 10,000 others via synapses. They connect and they send chemical signals to each other. Not only are there chemical signals, but there are electrical signals that are going back and forth as these connect. And each of these brain cells, you can imagine that each one of them is a bit like a snowflake. It's completely unique, but they're also, you can classify them in some way. So they're not completely unique. They have similarities between each other. And those similarities, I think, are important in trying to understand how the brain works. So if you look inside a cell, what makes each cell unique is what genes are turned on. And our genes, of course, live on our chromosomes. We have 23 pairs of chromosomes. We've got a set from mom, a set from dad. And on those chromosomes reside about 25,000 genes. Those 25,000 genes, which of those 25,000 genes and to what level they're turned on in each one of those neurons determines its properties, because DNA being turned on is part of a messaging cascade, which RNA then makes proteins, and proteins go about and do the business of those cells. So I'm going to tell you a little bit about a project that we have to map the human brain, to actually see what genes are turned on in the brain. Now our story starts, and this is, my story is deconstructing the brain in several hundred easy steps. We start at a medical examiner's office. This is a medical examiner's office in the state of Maryland. It's one of the primary sources for us to get human brain tissue. And it's basically the place where, if you have recently died and you haven't died in a hospital, you get brought into the medical examiner's office. It's a morgue. And so we have a team working. This is one in the state of Maryland in the US. We also work with one in California. And we have someone at the medical examiner's office who is screening for cases. And they're looking for our project, we want normal controls. Well, what is a normal human brain? I don't think anyone, probably many people would think, well, I don't have a normal brain. Many of you might say, oh, I'm completely normal. But we have very specific descriptions of what normal is for us. It's between ages of 20 and 60. We are looking for no history of neuropsychiatric disease. We actually do a complete toxicology workup to make sure that indeed there's no drugs on board. We have interesting things that happen along the way as we're trying to screen these processes. We've had a case where someone whose cause of death was listed as a heart attack. When the toxicology workup came back, it turned out that they had died of acute cocaine toxicity. So there's a lot of things here where we're very, very careful about getting the highest quality tissue. So when someone dies, usually they don't get into the morgue immediately, and the clock is ticking from the time a person dies. The tissue starts slowly degrading. And it's very important for the RNA that we want to read out that we have to make sure that we're able to freeze down the tissue within 24 hours after time of death. So you can imagine the orchestration that has to take place where someone comes in, there's a first screen to say this might be a good candidate for this, and then we have to obtain consent from the family. And this is another place where we often do not get consent. I think about half the time we get consent, and you can understand why. Someone has just recently lost a loved one, and someone is calling and saying, you know, can we take the brain for research? And it's a very tough thing. It's a wonderful gift for the people who do allow it. An interesting phenomenon, and one that has forced a bias in terms of the brains that we collect, is that women are much more likely to give consent of their husband's brain than vice versa. And so, people have joked with me about, you know, well, he wasn't using it anyway. But the reality is what this does is it affects our ability to get female brains. And so our project, in which we looked at 6 brains in total, 1 female brain and 5 male brains, and it wasn't, our bias is just in collecting the tissue, not in any other thing. So once we obtained consent for the brain, then we have to move very quickly. So the first thing that we do is we collect an MR. So this is something that magnetic resonance imaging, it's something that many of you, I'm sure, are familiar with. It's something that's done right here, but this is done post-mortem. So the body is actually wheeled into a clinical MR, and we take this. And the important reason for doing this before we remove the brain is that this becomes our first set of maps. This becomes the framework in which we're going to hang all of this other data that we collect. So it's a very important MR step to begin to hang the data so that people who are doing imaging on the living brains can come in and actually match up place to the other data that we collect. So we also collect something called diffusion tensor imaging, which allows one to see some of the wiring of the brain. And then the brain is removed from the skull. It's slabbed and cut into 1 centimeter thick sections, and it's frozen. So this is a frozen slurry. It's frozen solid, and it's shipped to us in Seattle for further processing. So this is one of those slabs. It's frozen solid, and it's in something called a cryotome, which is going to take very thin sections, cut them. It's about the thickness of a baby's hair, about the thickness of a little more than a neuron, a 25 micron thick section. And we cut these and we transfer them onto a microscope slide. And that's shown here. So we actually transfer these onto a slide. And then these slides are treated with stains to stain various aspects of the cells. This is an H&E stain, which is a very common stain used in histopathology. And then we take this information, we have them now on a microscope slide, and we scan it. We combine this with anatomic associations that we've made on the MR. And that helps us define how we want to further parcellate the brain. And the reason that we start to break this up into smaller pieces is that the large pieces are very difficult to handle. And I'll show you in a second what we're doing with these smaller pieces. But they'll fit onto basically a 2-inch by 3-inch microscope slide. So you can see here, this is cutting. This is a smaller version of a cryotome. This is frozen. And so it'll actually melt onto a slide. There's Da Vinci's paintbrush being used for a different task. And it's very carefully transferred here. Another thing to note is the barcode on the slide. We handle thousands and thousands of samples. And then we have robotics that do things like automatically stain this tissue, et cetera. So when we do these stains, we're able to get a lot of resolution. And really, these are guidelines for an expert team that we have at the Institute that tell us the various parts of the brain. So they know their way around, they know these structures, they can see various concentrations of cells and know that this is a particular part of the brain. That allows them to create these maps that tell us, map up those regions and tell us what regions we want to take as samples. So our lab technicians can then take that information and go in, and actually this is something called a laser capture microdissection scope. And you can see the little blue line there. That's actually scribing the area that they've just delineated. And that's happening on this microscope slide. You can see the little blue light there. It's actually cutting it. And there's a tube underneath that's going to collect that tissue. It's going to fall into that sample. And that's going to represent that particular part of the brain that we've collected. Okay? We now take that sample and we do some chemical processing of it, which basically purifies the RNA that's in that sample. So that RNA is representative of the genes that are turned on in that particular sample. And then we, through some chemistry, put a little chemical tag on that. And then we put it on a microarray. So if you've never seen a microarray before, this is basically the entire human genome spread out onto a glass slide. There are 60,000 elements here. You can count them if you'd like. But each one of these represents a different piece of the human genome. So there's 60,000 of those elements. Roughly, there are 25,000 genes in the genome. So more or less, we spotted at least 1 or 2 of these, which are called probes, per gene. Now, when we take a labeled sample, and this had received a labeled sample from a particular part of the brain, remember it's got a fluorescent tag on it now, and we do a process called hybridization. And then what sticks tells us the level at which that gene is turned on in that sample. And then we can assay all 60,000 of these and pull those gene expression measurements. So imagine that we've done this over and over again throughout all the different areas of the brain. This is just an example of an area that we showed before called the hippocampus, involved in learning and memory. And there we took 70 samples just from that region. And in total, for each of the first 2 brains that we looked at, we took 1,000 different samples in a variety of different regions. So now we bring all of this together. Remember, we're looking at where all these genes are turned on, and then we're bringing it back into this context of place. So what we always talk about is there's the where, and now we're telling you the what. What's turned on there. It gives you clues as to the underlying biochemistry of those regions of the brain, which are very important for people who study the brain. And we bring all of this into a suite of online tools that are available for free for anybody. If you had your laptop here, you could download this particular application to your laptop and look at all of this data. This is one of those brains that we just looked at. Remember the MR that we took? That's the MR. And we've also provide handy indicators of anatomic regions. And these colored balls that you see here, these represent the samples that we took. If they're green versus red, that means it's either turned on a lot, if it's very high red, or green, it's very, very low in those particular areas. So let me pause briefly and talk to you a little bit about where we've done this in the Allen Institute for Brain Science. So we are a not-for-profit research organization, and we were founded, as Gary mentioned, about a decade ago. And we were founded, it's not named for me, it's named for Paul G. Allen, who is one of the co-founders of Microsoft. Paul has invested over $200 million in the Institute over the last decade. Really, I think very excitingly, has given us this philanthropic resource to fuel and impact the field as much as possible, and that's really our mandate. So we're dedicated to making these tools and this information freely available for researchers around the world. And I have a slide that will tell you a little bit about the kinds of impact that we're having. As Gary also mentioned, we're structured differently than a typical nonprofit organization. We're structured much more like a biotech company. So I always like to argue we bring the best of both worlds together, where we have the discipline of approaching things in a very disciplined, team-based way, while at the same time we have this greater public good effort of getting everything out there. So unlike if you work for a pharma company or something where a lot of your stuff doesn't necessarily see the light of day, I think people that work at the Institute, such as the team here, really feel proud to contribute. And those teams are great. So one of the fun aspects that we have at the Institute is bringing people from a variety of different disciplines. So right now we have 55 PhD level folks out of the 225 staff. And those PhDs and skill sets range from PhDs in math and physics to molecular neuroscience, molecular biology, genetics, information technology. And again, the fun part is that we're all working towards these common goals. So we're not separated into departments and have our own sort of separate goals, but really collective goals together. And we are based in Seattle, Washington. As I mentioned, our overall goal is to understand how the human brain functions in disease and health. And our approach has been to create these foundational resources that scientists around the world can use. And if you wanted to think about the broader area of brain biology and the various areas which you could work, you might start on the left. And if you think about science in a reductionist way, if you think about taking apart the radio to figure out how it works, all the little individual parts, you'd start on the left, and then you'd work to the right. And on the right would be the program that you're hearing out of the radio. And so what we chose in the very early days of the Allen Institute, while we think these are great problems to think about over here, the reality is we really need baseline information to truly understand how the brain works. So we're going to start focusing on the left. And so over the last decade, we really focused primarily on looking where genes are turned on. We've looked in the mouse brain. We've looked in the developing mouse brain, the human brain. Now, moving forward, and I'll talk a little bit later, we're going to be starting to look at things like connections, dynamics, how the brain computes information. So our impact on our website, brain-map.org, researchers from around the world can come and access the data. So on any given workday, there's between 1,000 and 1,500 unique users of the data that come in, and they mine the data. They access it. 5% of the people that come in usually stay for an hour or longer using the data and the tools. And we know we don't actually require people to even register to use this data. Any of you could log in and access it. But we do know from a URL where these people are coming from. And so we know that scientists from every major pharma company use the data, from biotech companies, and then, of course, from academic centers around the world. And again, it's all for free. So We also know that researchers studying just about any aspect of brain biology that you can imagine are citing or using the resource in a variety of ways. And I've just listed, stroke, Alzheimer's, obesity, schizophrenia, autism, cognition, just goes on and on. So anything related to the brain, people are finding some value in these data sets. So since we started, you saw people transferring tissue to microscope slides. That's something that, even though we've got a lot of robotic manipulations to do, that's still done by hand. And 3.5 million tissue sections have been transferred to slides at the Institute over the last decade. Is a lot. They've been put on 750,000, 750,000 microscope slides. So that's 35 miles if you laid those end to end. It's a lot of microscope slides. And we image those down to 1 micron per pixel. So in 2012, just last year, we generated almost a petabyte of data. For those of you who don't know what a petabyte is, it's 1,000 terabytes. For those of you who don't know what the terabyte is, it's 1,000 gigabytes. It's a lot of data. One other aspect that we do at the Institute, we've been working on programs, which I'm not going to talk about tonight, making transgenic animals, transgenic mice. And our engineered mice have been distributed out to thousands of labs across the world. So how do people use the data? When they come in, they're mining the data. How could they possibly use it, and what could they find out of it? So I'm going to give you a couple of examples of things that we found out of this data, just as an example of the kinds of things that people are using it for. And the example here is just to show that I wanted to look at 2 different drug targets. So I'm not looking at the drugs themselves, and I'm not looking at individuals that have taken these drugs, but rather when you take a drug, drugs target proteins. They typically either turn up the activity or turn down the activity, and that's part of the way they act. And one of the important things that you want to know with a drug is where are its drug targets turned on. And so fortunately for 2 very well-known antidepressant drugs, Prozac and Wellbutrin, their drug targets are known. Not all drug targets are known. These are known, and there are these 2 genes here, SLC6A4 and SLC6A2. They're 2 different genes. And remember that both of these are antidepressants, but they have different side effects. And so it's a very interesting thing when you start to look at these areas where you can see similarities, things where these genes are turned on, and then other areas where they're actually different. And so for people studying antidepressants, wanting to understand what the range of side effects are, these are very important clues to start to understand what that side effect profile might be and how they might start to work around it. Now, another thing that you can do with these is actually do something like a database search where you can say, I like the profile of this, show me everything else, every other gene in the genome that has a pattern like this. And for the first time, you can actually do that. You can see and find a whole collection of novel, potentially novel drug targets. It's certainly over the last decade been the era of genomics, human genome sequencing. You'll hear a lot of what people call genome-wide association studies. These are large-scale studies in which people's genomes are being sequenced, or at least a high degree of information about their genomes is being pulled out. And they're trying to find genes that are associated with disease. Okay, so when you do a study like this, maybe you're interested in schizophrenia, you enroll 10,000 people, and 1,000 of them have schizophrenia, and there are other controls, and you're looking for things that correlate with it. So if you have the disease, do you have this particular genotype, if you will? So what you get out of those studies are lists of genes. They don't tell you anything about the where. They don't tell you anything other than a list of the genes. And so this becomes a very important resource for those people who are doing those studies on people with brain-associated diseases to go in and say, well, here's a collection of genes. I know they're interesting to the disease that I'm looking for. Let me get some additional clues as to where those genes are turned on. And so this just gives some examples of the kinds of things that people can find out of the resource. So we talked about before normal the normal brain, and what is a normal brain and what is a unique brain. And so I think most people would agree that they're unique. The humans are different. This is the diverse workforce of the Allan Institute. I'm hiding in there somewhere. See if you can find me. But we were very interested in understanding what the differences in the brains that we were looking at were. What are those gene expression differences across different individual brains? So for the first two brains that we looked at, we were very interested in this for a whole series of reasons. One, we wanted to make sure that we were producing high quality data. We want it to be reproducible. But the second was a very fascinating one, which is, what are those similarities and differences among individuals? They're interesting, but if we want this to be a baseline resource for people to use, we want to know that there's a lot of similarities and few differences. And indeed, when we looked at this is looking at 150 different regions and over 500 genes. There's a great deal of correlation here. I know that showing data plots sometimes, this is very simple. Everything in the cloud here that's on that line means that it's similar in this brain versus the second brain. These are completely unrelated individuals. They live separate lives, and remarkably very similar correlations here. Now there are differences, and those differences are unique and important. So those things that are hovering outside the cloud, they're things where they might be low in this individual but high on this individual. We actually have done some other studies where we've looked using a process called in situ hybridization. Now, this is a different process which tells us where a gene is turned on in a cell, in a tissue. And what you see here, if you're looking carefully, you can see little black spots. Each of those little black spots is a nerve cell. And those nerve cells are turning on that particular gene we're looking at. So there's lots of lighter colored spots, and then even spots you don't see. There's lots more nerve cells in there, but the ones that are turning on that gene are dark colored. And so this is an example, two examples of genes in which, back to understanding this, what are those differences among the individuals? They're likely to be these things which are very specifically turned on just in a specific subset of cells, that cell class, that cell type. So for example, relin, which is involved in neuronal migration, it seems to be just turned on in this layer 4 in this first donor, but not in these other two. And same thing for this disc 1. There's just layer 1, 2 in one individual and not in others. Now, we did a study on the mouse several years ago where we looked at a variety of different strains of mice. And these were mice that were varying degrees similar or different to each other evolutionarily. Their genomes have been sequenced. These are well studied in the lab. And so we know that things on the left here, at C57 Black 6, which is a standard lab strain, versus Spreadus, which is a wild-derived strain, which is the furthest related. And what you see is that a similar pattern where across different species, across different subspecies, given a particular gene in a particular structure. And in these cases, this is all dopamine, which is a very well-studied neurotransmitter, dopamine receptor, DRD2, or tyrosine hydroxylase, which is involved in the biosynthesis of it. And what you can see here is that in some of these subspecies, it's turned on a lot in these structures, for example, here and here, and in some it's not at all. So again, there's this theme that there's these individual cell classes or cell types which seem to be important in whether or not these things are turned on, and is probably an important driver, especially when you think about evolution, when you think about what evolution might be acting on. And for example, here, when we do a comparison of this across the further out you get in terms of evolutionary distance, the more of these discrete changes in cell classes that you see. Okay, so I'm going to switch gears now and talk a little bit about the future of the Allan Institute, but I think even more broadly than that, the future of neuroscience. So I hope most of you would be familiar with something which is termed the central dogma of molecular biology. This is something that probably about 50 years ago, there was this really golden period, and it wasn't very long in terms of history, where very rapidly there was the discovery of DNA, and then really the rules behind how DNA encoded RNA, and RNA then had this triplet codon message thing that told the cell how to make proteins. Okay? This is incredibly important. You think about all the impacts that's had on molecular biology, et cetera. Now, perhaps coincidentally, that was 50 years after a similar set of discoveries or principles was elucidated for the atom. And you think about the understanding of chemistry and quantum theory. And so it's been 50 years. And I'm going to propose that we're on the cusp with all the technology advances of understanding the language of the brain, truly how the brain computes information at the cellular level. And I believe this is all anchored around this idea of a cell type. Okay? And understanding those cell types and how they behave, what their underlying molecules are, is going to be key towards understanding this language. At the Allan Institute, we're embarking on programs to start to look at this in a variety of different ways. And we're looking at both mouse and human because they each offer interesting advantages to study. In the mouse, we're going to be looking at detailed cell type descriptions and functional descriptions, and the same in human as well. So let me briefly talk about what I mean when I say cell type description. So I've just talked about gene expression, looking where genes are turned on in a cell. But you'd also like to understand, and this is something we can do now on individual cells. So before I showed you something where we're taking a laser and we're cutting out a region, now we can actually separate individual cells, and you put them through a little flow sorter, and you can read out genes, about 100 genes at a time, in a very quantitative way, what genes are turned on in those individual cells. And you could do this over and over and over again. So we're able to now do that, and we can do that for both human and mouse cells. You can also read out their electrical properties. So you can stick a little electrical probe in there, and you can start to read out what their electrical properties are. So now you can start to link what genes are turned on and their underlying biochemistry with the patterns that they show with their electrical activity. And you also want to understand their morphology, how they're connected up with others, as well as their projections, how they project out. So for understanding function, the mouse is a very, very great model for doing this. And we're going to focus on the mouse visual system. And the mouse is a great model system because we can actually look in real time at what the cells are doing and how they're firing. So there's a great set of tools that's been developed over the last several years which allow you to, as this animal is going through a paradigm, he's looking for a certain sort of thing, and when he does, he gets a little reward of juice there. And you can actually look at the cells as they're firing in real time. And you can see what cells are firing and in what order, and understand their electrical properties in addition to how they relate to this function and this behavior. And now you can connect that up. Once you understand that, you can then take that tissue and you can do electron microscopy, and you can actually see how those individual neurons are connected to each other at a very fundamental level. So remember I said that each neuron might be connected to up to 10,000 others. So it's a daunting problem, to be sure, but it is a solvable problem, especially if you're willing to apply industrial high-throughput ways of approaching this, where you want to go fill out a matrix of this information. You want to go systematically, cell by cell, and start to put all of these pieces together. And then, remember, link them to these molecular properties that we just talked about. So how do we do this in humans? We do this in cells. Okay? So it shouldn't be any surprise to anyone, hopefully, that stem cells are an exciting new world for the study of humans. So from any one of you, we could take a skin pinch, just a little pinch of skin, get some fibroblasts, add a few factors in a dish, and we can get them to go back to a native state. And then in that native state, we can then drive them to make neurons. We can have them make neurons in a dish. We can do this here. So we can do this for hundreds, and ultimately we'll be doing this for thousands of individuals. So we can make those neurons. Now, they're not going to function the same way they function in a normal human brain, but they're going to have a lot of the fundamental properties. They're going to turn on the same sorts of things. And we already know through some pioneering work that a number of people have been doing very recently that if there's somebody who has a particular mutation, they know that they have a certain disease, that you can actually see parts of that recapitulate in a dish. Okay? Testing those functional properties. So now we're able to do that, and it's a really remarkable thing. The other thing that we can do is we can engineer. We can take those cell lines, and we can actually engineer. We can put reporters in, or we can start to make specific genetic changes to start to test hypotheses. So one of the big challenges that we have right now in human genetics is sorting out this huge collection of genes that all seem to be contributing small parts to complex diseases. And trying to sort that out is hugely challenging. But the ability to engineer in the dish allows us to be able to start to test those hypotheses in a variety of different ways, and to do it in using these sort of industrial techniques. Again, very powerful. Of course, we can do single-cell measurements. And then finally, we can even do things like I just showed you in the mouse. This is calcium imaging. This is neurons firing. They're in a dish. There's a number of them. They've connected up into a rudimentary network, and they're actually able to fire and give action potentials. So we have the ability to start to do these functional tests in the human. It offers a great opportunity for us to really start to make some headway and to understand this. Now, the final piece is something that the Allen Institute's always done, is to share the data and to bring it together as a larger community. I'm here as part of this celebration for opening up the INCF node here in Australia. And this is the international community that shares this data, that pulls us all forward. It relies on the global community for helping solve really, really hard problems. And for us, it's no different. The data that we create, we want to create these frameworks for open sharing of this data and to get lots of people involved in looking at it, because somebody's going to figure it out. Somebody's going to put it together. You know, we, of course, we're happy to contribute to it. We know it probably won't necessarily be us. It'd be great if it was. But people will be solving these hard problems together. And so, you know, the technology is now, I think, ripe for, you know, with the ability to share lots of data for high throughput computing. And I think it's the time to be able to do this well. So this is how we bring it all together. For us at the Institute moving forward, we're looking at the mouse behaving in the whole brain. We're building a parts catalog on brain cells. And then ultimately, we're tying this in with development and disease in the human and being able to test specific hypotheses about how the human brain works. And we have a long ways to go. It's been a long time since Da Vinci. If you think about all the technology changes that have happened since that time, and then think about all the ones that are coming forward, I think it's a great time to be alive. It's a great time to be a neuroscientist. So with that, I'll thank you.
Thank you very much, Allan. That was absolutely fantastic. First, I'd like to acknowledge all of the sponsors of the forum: the Bionics Institute, IBM, Bio21, Bionic Vision Australia, the VLSCI, Melbourne Convention Bureau, NICTA, CSIRO, RMIT University, Monash University, Melbourne Health, the Centre for Neural Engineering, St Vincent's, Melbourne School of Engineering, Phillips, Ormond Fitzpatrick and ANSTO are all of the sponsors of this forum and of other events which the forum will be holding throughout this year. Please join me again in thanking Allan Jones for a fantastic presentation.